Hello and hola to everyone. Welcome to our 48th session. My name is Sang Ni, and I am one of the organizers of the Porous Media Tea Time Talk and Interpol Young Academy. And today I have joined here with me the rest of the team members. We have Ramin, who are a recent addition to our team, and I believe this is his first appearance in our streaming, so welcome. And we have Muhammad and Mahdi. Today's session is a special session where we invited two great speakers from Brazilian Synchrotron Light Laboratory who will talk about how we can use synchrotron series to porous media research. So I'm going to introduce our first speaker, Dr. Daphne Pino. She is a geologist specialized in hydrogeology, focusing on fractured aquifers and groundwater contamination at regional and local scales. A couple of years ago, she also started studying porous aquifers at microscale, which led her to current work at Brazilian Synchrotron Light Laboratory. Please welcome Daphne, and floor is yours.
Thank you. Let me share my screen. Okay, so hopefully now you can see my window. Yeah?
Yep,
okay.
We do.
So thank you everyone for being here today. I'm Daphne, and I'll be speaking to you about cleaning up contaminated soils and aquifers, and we're focusing on investigating pore-scale processes with synchrotron X-ray microtomography. So this image that you see here on the right, this is the Sirius laboratory, which is our synchrotron light source here in Brazil. So why am I talking about groundwater? Well, one of the Sustainable Development Goals from the UN is access to clean water and sanitation. So I think this explains all. Like, we all understand the importance of water for life. So when we're talking about Brazil, we're talking about over 200 million people. And this is a country that had the latest GDP in 2019. So it shouldn't be, we shouldn't be in such a bad situation. That's what you would think. But however, 35 million people in the country do not have access to treated water. And when we look at all municipalities in the country, 52% of them depend on groundwater for water supply, and that's either they are entirely dependent on groundwater or partially dependent, which means they use both groundwater and surface water. So this is clearly a very important resource. So just to give you an example, here you see the state of São Paulo, which is where I live, and here we have the metropolitan city of São Paulo, which is one of the most industrialized areas in the country. So here on this map on the right, hopefully you can see these darker orange areas and this red one. They are all areas in the city where you have industries concentrated. And all these black dots that you see here in this map, you see a lot of them are on the top of these industrial areas. Those black dots, they represent deep supply wells. So those are wells that are extracting groundwater for different types of consumption. And why this is a problem? Because when you're doing this in an industrial area, you have a great chance of, as you're exploiting groundwater, you're lowering the water level that you have in your aquifer, and you're probably bringing any contaminant spills, any plumes of contamination that you have on the surface, you're also bringing them downwards with your water level, and you're contaminating other levels of your soil, and you're probably spreading this contamination through your aquifer. So this is a very big problem in many industrial areas, in many urban areas, not only in Brazil, but throughout the globe. And we did some studies here in this red area that you can see here. So this is a typical study that we do as a geologist that you see at the regional or local scale. So we usually go with a driller. We have our workstations here in the field. This is a very different scale from the micro scale. So you see me here very happy with along rock core, we usually work with soil and rock samples. We have stations for preparing those samples. We were studying here our vertical profile with different soil types and rock types. We studied outcrops. We did geophysical profiling in as well. So all of this is a very different scale that usually geologists are used to that, because we're usually looking at this local scale or even larger scale, even regional scale. But what is controlling everything that we were studying here is actually what is happening at a micro scale, what is happening at a fracture or at a pore scale. And to study that, we cannot just gather data directly from fieldwork, but we can use synchrotron light. So what is synchrotron light? It is a type of radiation that has a high brilliance and high energy, and it has a broad spectrum, because you can have synchrotron light from infrared to X-rays. You see that the visible light is here in the middle. So the type of synchrotron light that you're going to use, the wavelength that you're going to use, it will depend on the kind of experiment that you want to perform. The two examples that I'll show you today, they both use X-rays. And this is also the Beamline that we work at Sirius. It's working with X-ray microtomography. So this is another photo of our lab. So as I'm talking about groundwater, something we want to do, as we have seen clearly, that contamination is a huge problem. So one of the things that we want to do is clean up. That's the title of the presentation. So this is what we call a remediation process. So here is an example of a reaction of a solvent. This is a very used industrial solvent. It can be used for cleaning metal parts. So it's used in many types of industries, and it's also one of its father products. It's also used for dry cleaning, for example. So it's a very common contamination in urban sites. So what has been shown over the past decade or over the past 20 years is that this type of solvents, they react very well with nanoparticles of zero valent iron. And that's the reaction that you see here. So here you can see some images in the microscope of those nanoparticles, and they are usually presented as powder. And what we want to see is how exactly these reactions are happening in a microscale. So this is an example of a paper that our group published. What you see here in red is the solvent, the TCE, and then we injected nanoparticles. And after the injection of nanoparticles, what we saw, this yellow part here, is actually gas that was produced during the reaction. And we saw the consumption of TCE, but we also saw that this gas that was produced, it was also dislocating some of the solvent droplets. So this was very interesting, and this could only be done using synchrotron light, not only because we had to see things at a very small scale, but also because we would be able to image the process at different times. So in a synchrotron beamline, you can also have images at different times, so you can almost have something, you have resolution in time as well. You can almost make a movie of what is happening in your sample when you have an injection experiment. So what basically happens in the beamline, we have the X-ray source. So we're going to, I forgot the word in English, sorry for that. This radiation will go through our sample here. Our sample, it will have to rotate so we can actually get different size of our sample. So later we can reconstruct a 3D image. And based on the composition of the sample, we'll have different absorption coefficients. So the sample will absorb or let the radiation pass through it in different ways, depending on its composition. And the radiation that comes through the sample will be collected by a detector. And this detector will provide information for us to later analyze those 3D images and separate phases and understand what is going on with our sample. So what we did, the first experiment that I want to show you is based on the nanoparticles injection. So here, this is our flow cell. So we were injecting things from the bottom and going through here. This would go in front of the X-rays. So X-rays would go through the sample when we would, let me show you on the side, it would go through the sample when we would collect the data on the detector later. So we had a sample with beads, with glass beads, and then we filled it with water. So we would have a first image of that with our sample, with our grain, with our porous media, with water. Then we put the sample to 2 rounds of nanoparticle injection, and each round of nanoparticle injection was followed by a final water injection. So we had a sample saturated with water, then injected nanoparticle, water again, then nanoparticle again, and finally water in the end. And what we wanted to see what was controlling those nanoparticles at a pore scale. And something interesting about this experiment is that we were imaging this sample. It's about 1 centimeter long. We were imaging in 3 sections, so we had better resolution for a long sample. So if you never saw one of these images, this is about what you got. It looks a lot like a radiography. This is a tomography image. So we see water here in the darker gray and our grains in the light gray. This was after the first water injection, so that's all we have. This example is after the second nanoparticle injection. So you see here, there's water, nanoparticle, and water again, our 5 injection phases. So we have, again, the grains. We can still see the water, but now we see the nanoparticles here and a brighter gray. And we also see that some gas bubbles were formed here. So this is the kind of image that we get from our detector. And what we do, we're going to segment, we're going to separate those phases. So we can calculate volumes, you can calculate surface contents, basically anything you can think of. And we can also perform permeability experiments once we have the data regarding porosity on each one of them. And what is very interesting here is that we see after the final water injection, we can see that some places that before they had nanoparticles, they now were filled with water. So we could actually see that the water we were injecting was dislocating some of those nanoparticles. And as we expected, those nanoparticles, they are usually retained in smaller pores, like the portholes after the final water injection. But what I really want to show you on this experiment is this graph over here. We have the pore space, like percentage of area of pore space, so our porosity here, and the slice, so the bottom of our sample and the top. So after our first water injection, we had between 40, this is the light blue that you see here, this one, we had between 35 and 40% of porosity. After we injected nanoparticles, we go through this red line here, this yellow line. So we lowered our porosity between 15 and 20% through our sample. So this is kind of what we were expecting, and we're happy because the nanoparticles were actually filling the pores, that's what you want to do for injecting the field to react with a contaminant. Then we injected water again to wash away those nanoparticles, and we succeeded because this is what you see here in this medium blue here. We basically recovered all our porosity, so we're able to wash most of those nanoparticles away. Then we said, what happens if we inject nanoparticles again? What we will look like? Then we got this orange curve here, and we see we actually reduce even further our porosity. So what we think that was happening here, either we were able to inject more nanoparticles, or what we believe that was happening, especially because after we injected water again, we actually get this dark blue here. So we see we did not fully recover our porosity after the second nanoparticle injection. And that's what we believe that happens here, is that nanoparticle history is very important. So while it looked like at the first time we actually were able to wash all nanoparticles away, that's not true, because some of the nanoparticles were actually retained in our media. And those nanoparticles that were still there, they were reacting with the new ones, and that was causing more clog in our system. And this kind of thing is very important because when you're doing this in the field, you cannot see this, and you might be clogging your media, your soil, and the nanoparticles that you're injecting, they might not reach the place that you need to remediate. So this is very important to consider when you're going for a field scale injection experiment. The other experiment that I want to show you very fast is for an in-situ chemical oxidation experiment. So here we have again the grains and the water, and here we actually injected the TCE here, we injected the contaminant. And lastly, sorry for these two, they are filtered, but this one is not yet. But after that, we injected permanganate, so we injected an oxidant reagent. So we can see that there is a lot less solvents here. This is still an ongoing experiment because we have students working on this. And we're still working on what's happening here. But one of the interesting things here is that we saw very, very important variations of pressure in our system. So we're investigating these reactions, what is causing this variation of pressures, because this could be very significant depending on what you're doing also during field work. And we can do the same kind of analysis of distribution of porosity and permeability here. So what I hope you remember from this presentation is that with synchronous light, you have a very powerful microscope because you can see things at a micro scale, and sometimes, depending on your experiment, even at a nano scale. Microtomography is the non-destructive method, so you can have you can study your samples, you can take smaller portions with higher detail without destructing your samples, and you can do studies at a pore scale. And we've been doing these studies of dynamics of fluids in porous media because, as I said before, the microscale is a reflection of what is happening in the microscale, and that's why this is important for fieldwork studies, for fieldwork remediation processes. So we had the example of transport and delivery of nanoparticles in porous media, and a quick pass through the chemical oxidation processes at the pore scale. And all of this is because these things, they bring insights for flow models in remediation studies, because we often have a whole remediation project, and it happens that sometimes it doesn't work exactly like we were hoping for. So it's very important to understand these particle mobilities and entrapment mechanisms. So if you want to know more about cities, I invite you to visit the webpage here. You can find more information on the lab, and there is also a call for proposals. I think it's coming up in September, so there should be information there soon. So if you have a research that you think you could develop it in one of the Beamlines, you could also apply for that. And if you have questions with that, we can help you later. I want to thank you all for being here, for hearing this talk, and for our partners in the nanoparticle project. And thank you again. If you have any questions, I would be very happy to answer.
Okay, now we have to hand it to Ramin to ask any question if he has, and also take care of the questions from the YouTube. So Ramin, go ahead.
Yes, thank you, Daphne, for the very nice talk. Very interesting. I actually have a question before going to the comments. So I was wondering, just curious to know that if you have any plans to basically study other type of contaminants like PFAS, microplastics, pesticides, and those things also in groundwater, because I assume that those also exist. Yeah.
Those are very important contaminants, especially PFAS. They're like the hot topic right now in groundwater. We choose first those chlorinated solvents like TCE or PCE because we were actually interested in understanding the interaction of nanoparticles in the porous media with those solvents. But we don't have projects like right now for this, but I myself, I would be very interested in working with microplastics and PFAS. As a hydrogeologist, I would be very happy to work with that. So if anyone wants help with projects, just let me know. I'd be very happy to work with that.
Yes.
I actually have two questions, if I may.
Yes.
I personally work in remediation researches, so I'm very interested in your research and talk. So my first question is, what happens to these nanoparticles after they react with TCE? Do they become dissolved, and we don't have to worry about their aftermath? What happens to them?
When you're in the field, you mean, right?
Yes.
They are zero valent ions. So what what happens is that you have iron ions in your groundwater later that might have to precipitate. So that's the usual balance that you have to do. Do you prefer to enrich your aquifer in iron, or do you leave your solvent there? Well, that's usually what's going to happen. So can I share again? Let me see, share the screen. If we go back to the reaction here, so this is what we'll have. We had the TCE, and you have the zero valent iron here. So you will be left with 18 that's like a lot less harmful than TCE or any of its daughter products. But you also be left with some iron ions.
Mm-hmm.
So yeah, it's not a perfect thing in my opinion, because you're still enriching your aquifer with iron, but that's still way better than having TCE on your aquifer, because if it degrades to DCE or to vinyl chloride, it's even more toxic than TCE because it can cause cancer. You have trouble with vapor intrusion. You have a lot more issues. It's a lot harder of filtering, of taking that out of your groundwater than the iron if you want to later use your groundwater.
Okay.
So I think it's a balance of what is the worst that you can have in your groundwater. But I understand the worry. I usually have this kind of concerns as well.
Thank you. I have quick second question. That if you inject this kind of nanoparticles into porous media or fractured aquifer systems, I think you will have preferential flow paths, and there will be some contaminants that are trapped in less permeable areas. So how do you plan to target the contaminants that are sort of trapped in those regions?
Okay. We have been doing mostly work. We haven't gone to field scale yet, but there are works, I think, especially in Czech Republic, you can find some works that they have been applying in field scale. So there are things that you have to consider before injecting them. For instance, if you have too much clay in your media, because you can have a sandstone with different degrees of clay in it, of cementation, that could damage your injection because you won't be able to inject much of those nanoparticles in there. So you have to consider if your aquifer, if the composition of your aquifer is able to receive that kind of injection, because it might not be the best suitable option for your case. So it's not just because they react very nicely with your contaminant that this is the best that you can use, because that's exactly what we're doing, why we're trying to understand how they move in porous media. And we're starting with a very simple system, actually, because we're just working with sand grains. They are not consolidated. So it's still a bit different from what we see like in the actual aquifer. We do our best to mimic, but it's not the same as having sandstone with clay and many other minerals. So you have actually column studies that you can also try that. You definitely have to consider maybe you would need different injection wells at different depths. That could also help. It would be very case specific depending on what you have on your site. And regarding the fracture media, you never have an homogeneous flow in fractured media because you're always going to have preferential flow paths. So for that, you really need to understand your fracture network before injecting anything, because you can. That's even an even harder response. I don't think I've seen studies of nanoparticles in fractured rocks yet. Most of the ones I saw, I'm not saying they don't exist, but most of the studies out there, they are in porous media. But there are also some softwares that you can, like one developed by the people of the Polytechnic of Turin, that you can simulate the injection of nanoparticles in the porous media, and they are going to give you information on this microscale, because they bring their data. You can also input data from column tests. So it's a very nice software. Could be very useful it's very useful for building this bridge between the lab work and the field work. So there are different tools that you can try to do the best thing that you can for your field work.
Thank you.
Yes, I don't see any questions in the comments. If anyone else has a comment or a question, Mohammed, Mahdi, or no? Then I would ask Sang to introduce the next speaker. And we thank you again, Daphne, for the very nice talk.
Thank you.
So I think, Mohammed, can you go ahead and introduce our next speaker? Oh, actually, I'll do it. Just give me 1 second. So our next speaker is Dr. Aluizio Salvador. He is a physicist graduated from the Federal University of Paraná, Brazil. Currently, he is a postdoctoral researcher at the University of Sao Paulo, acting at a Brazilian synchrotron laboratory in the city of Campinas, Sao Paulo, as a collaborating researcher, working on ways to visualize and understand the process involved in the capture and storage of carbon dioxide by rocks. Aluizio, floor is yours.
Okay, thank you, Sam, for the introduction, and thank you all the team for the invitation to be here. It's a pleasure to be here. And I just want to add that I have a FAPEP scholarship. You can see my mouse, please. You can see my mouse in moving or not?
Yes, we can.
Okay, so I just want to add that I have a scholarship from FAPESP in collaboration with the Research Center for Greenhouse Gas and Innovation. And today I want to talk a little bit about the efforts and also the challenges we are facing in the development of a microfluidic device using a real rock as the sample, where we wanted to see the process involved in the mineralization process, the mineralization of carbon dioxide, by using synchrotron techniques. Specifically, we're going to analyze rocks from the Paraná Basin, which is located mainly in the south of Brazil, but it also spans to other countries. In this map, we also have in the yellow dots the ethanol and sugar plants that are believed to be the main source of carbon dioxide emission. So this good source sink matching would be easy to transport the carbon dioxide from the source to the sink, and it has encouraging a lot of characterization, reservoir characterization about these rocks. These previous works have shown some previous good results as well, and the reservoir presents a good porosity and permeability that would allow the injection and the storage. There is a sweep of depth for geological storage in this basin, and mainly it's formed by basaltic rocks, which would be ideal for the mineral traffic. However, there are yet many studies to be performed. This base is very heterogenic. It's very big, so we need to map it. We need to sample many parts of this basin. There are faults breaks in the seal layers. It would be a problem for the injection because it could cause leaks of carbon dioxide. And aside these very interesting properties, there are only few studies that are yet on the literature, so we need yet a big effort on characterize these rocks. Given this information, the goal of my postdoc project is to make a high-end characterization of these rocks, mainly evaluate the potential of this basin to capture and to store carbon dioxide. We are planning to do it by using all the research of the Brazilian Synchrotron Light Laboratory series. Daphne, I will talk a little bit about, but I will make also a brief introduction about the series and the potential. Focus on two beamlines that we are going to use, the Carnaúba and the Mogno. Also, I will talk about our rock-on-a-chip device that we are developing, and then I will let my final comments. This in the picture is the Brazilian Synchrotron Light Laboratory called Sirius. Here we have the parking place where it's possible to have an idea of the size of the Sirius compared to the cars. In a synchrotron light, electrons are inside a ring. Here in the Sirius, the ring has 500 meters of circumference. And strong magnets are responsible for keeping the electrons inside the rings while it rotates. But when the magnets bends the ring, the electrons to be kept inside the ring, it generates the synchrotron light. These synchrotron lights account for a wide range of the electromagnetic spectrum, and it can be selected and directed toward the beam lines along the ring. The advantages of using a synchrotron light is the high flux. We can make images of small parts of some structure and with higher resolution. This is the largest beam line of the series. It's called the Carnaúba that I'm going to introduce now. It has approximately 145 meters between the source, where the synchrotron light comes from the ring, until the experimental station, where the sample is analyzed. Along this path, there are set up with mirrors and slits and monochromators with the goal to demagnificate the synchrotron light so it can reach the sample with a nanometric beam spot. That's why this beamline is called a nanoprobe. That's why this beamline has a nanoprobe. In this beamline, you can study many types of nanostructural materials by using 2 or 3-dimensional images with X-ray absorption, emission, diffraction, or optical emission contrasts. However, to make our experiments, our mineralization experiments available in this beamline, we need a big effort on the sample preparation. And here is where the microfluidic devices take place. Just for a contextualization, microfluidic devices are known to operate with small amounts of sample and channels that can vary from micrometers to submicrometers. Also, microfluidic devices have low size, low weight, but a high throughput, and that's why called a lot of attention from the synchrotron communities to perform in situ or in vivo experiments in the beamlines. As an example, microfluidic devices have been also applied to understand and study pore medias. Here is a pore surface that tend to mimic a subsurface of a rock. However, these micromodels have an inherent problem. The engineered materials used to print this model, like silicon, glass, or PDMS, they do not fully represent the chemical reactivity of the rocks during an experiment. So, for example, in my case, I want to see the reaction of a solution and how it would make a mineralization process available. I cannot use it on a micro model, for example, with engineered material. So what the scientists have made is using a real rock as the sample, not an engineered material, and that's what I'm going to show in the next. That's a rock on a chip or microfluidic device that we developed specifically for use in the Carnot beamline, using the non-spot of the Carnot beamline. Here's just the project. So we draw a channel in one of the surface of the rock. This channel has 1 millimeter wide and 0.5 millimeters deep. At the end of this channel, we have 2 holes with 1 millimeter of diameter that will work as the inlet and outlet of the channel. Here in the right, in the pictures, so we show our sample, or real rock, with 5 millimeters of thickness. In one of the flat surface, we have the channel draw with the 2 holes. And from the back view, we can see the inlet and outlet of the fluid. So the idea or the goal here is to use a peristaltic pump, so we can inject the selected solution to this part, and we have a controlled environment to analyze the mineralization and the results of the interaction between the solution and the rock. So these are primordial conditions to use a beamline to perform experiment in a synchrotron beamline. You need to have a controlled environment, so we can compare measurements before and after experiment. Complementary to the measurements we're going to do in Carnaúba, we are also going to use the Mogno beamline. That's the one that Daphne showed the results of her work. So in the Mogno beamline is specifically dictated to tomography, microtomography images, and it has very specific characteristics, mainly because it's cone beam geometry that I show here in the picture. So with this cone beam, we can perform analysis in the same experiment with different field of views, looking to different parts of the sample just by changing the position of the sample along the beam. So we can go from fields of view or areas of interest of millimeters to micrometers very fast. Also, with a full capacity of series, we can perform tomography images in seconds. And as Daphne also mentioned, we can follow in time determined reactions, having many tomographies for each 10 seconds, for example, and to see the evolution of a reaction that for us, specifically the mineralization process, where we want to see the microstructures change induced by the carbon dioxide, would be very interesting. And finally, in the Mogno beamline, we can make measurements with different energies at the same time. So as Daphne also mentioned, I'm taking many parts of her presentation. So the results depend on the coefficient absorption of the material. So if you have materials with different coefficients and interact with different energies, we are going to have different contrasts, and it helps a lot to differentiate phases inside a sample. However, due to its characteristics, we cannot use the same device that we use in the Carnot beamline. So in the Mogno, we are going to use this flow cell. This flow cell was developed here by the Mogno's team, and it uses nylon filter and beads to hold unconsolidated samples inside this PFAN tube. So this tube is transparent to X-ray, so it allows us to perform experiments to fully rotate the device to make the tomography experiment. Also, we can inject fluids through this flow cell, so we can follow in time, as I said, a specific reaction. And what we are planning here to make an experiment as close as possible from the one that we are performing at the Carnauba beamline, we will substitute these non-consolidated samples for a cylinder tube. The cylinder will be inside this tube, and we'll draw a channel along this cylinder. So when we inject the fluid, it will preferentially pass through the channel instead of the pores. So again, we are going to have a controlled environment, which for us that are at the beginning of the project, it would be very important just understanding how the surface reactions happens here. So, all right, going to my final comments. It is the main purpose of this webinar. I'm also going to talk about the challenges, aka what went wrong. And the first challenge started at the very beginning that was to obtain a clear cut of the samples. So we got the sample as removed, as collected from the field with non-defined shape. And we first needed to get slices with the 5 millimeters of thickness. And it was already quite challenging because we don't want to involve a process with chemical lubricants or resins or glues to fix it, because we are going to make chemical analysis. So, and chemical lubricant would be contaminate our sample, so we wouldn't be able to see the reactions that we want. Also, we want to avoid the polishing process on the surface for the same reason of contamination. So the cutting needed to be as precise as possible to obtain a flat surface, so the Kapton tape would make its work on sealing this channel and avoiding the leaks of the fluid and keeping the fluid only inside the channel, which, by the way, was our second challenge, to draw the channel with a relatively very small channel, 1 millimeter wide, 0.5 millimeter deep in the rock. As you can see, it was not all sunshine and roses. We broke many samples before we get the ideal conditions to obtain this microfluidic device. I think I mentioned it by the presentation, so this is just the beginning of this project. These slides are the results of our 2 or 3 months from now on, since the beginning. We didn't have the opportunity to use the device and the beamlines yet. Hopefully, on next month we have time on the beamline to perform the first experiments and validate it. And here it is, little move. As just show the rock, the channel of the rock. And here is the peristaltic pump we are using. The channel is upside down there, and we use a flow of 0.5 milliliters per minute. So this low flow is to allow the solution interacting with the surface in the channel for as many times as possible without the solution go too deep in the pores, which now is not our interest in understanding how the solution reacts inside the pores, but just in the surface. And we saw it, the Kapton tape did its work in the ceiling. No, we didn't have leaks. And now the next steps, besides to validate the microfluidic in the beamlines, is to improve the device by considering temperatures to control the temperature in the reaction, as well pressure, and try to reproduce the conditions as close as possible what we have in the nature. Well, I think that's it. Like Daphne mentioned, you all can apply for use the Synchrotron facilities. You just have to access the site. You have all the following rules there. And of course, if you need anything, you have so you have a question about the beamlines and the requirements to use these, you can contact us as well. Well, thank you. And if you have any questions, then please to answer.
Thank you, Aluizio, for this nice introduction to the development that you are making. And it certainly would be interesting to see some results out of the interaction of fluid with the rock properties and rock surfaces. I have a question of my own, maybe it's a naive question, but what are the time scales and length scales that you are expecting to see reactions or interactions of fluid with the surfaces in the synchrotron facilities?
Yeah. Well, as I said, we have a capacity both in the cardinal UBA and Mogno beamlines to see nano alterations. So as we are planning short experiments, like 7 days for mineralization are related short experiments, so we are expecting to see that like nano changes, like nano mineralization, nano precipitations, or changes in the nanostructure of the pores, not much as that.
Yeah. But do you have any idea about the time scale of these reactions?
No, I mentioned that we are like in the very beginning of these experiments. I didn't show here, but we start with the bench experiments, the bench test before to go to this beamline. And we performed experiments with 2 weeks, and we couldn't yet see the results that we are expecting. So we are not thinking on increase time, but we are thinking on the contrary, to decrease the time, but change the conditions to accelerate this process. Because like we don't have 1 month available at the beamline, so we needed to accelerate this process to make this experiment like 1 week the most.
Yeah, excellent. And the second question is just more of a broad question. Maybe both of you can answer, but we are starting with you. If anybody wants to collaborate on this research, either with you or with the serious synchrotron facility, what are the possibilities for the researchers based in Brazil or worldwide, around the world?
Yeah, I think the short path would be to contact a researcher here in the series or the coordinators of the beamlines, show the interest. And there are some possibilities, like you can be associate research, as I am here, because I'm a student from a university in São Paulo, but I'm here as a research collaborator. So if you are in other university or other place, you can act as a research collaborator, spend some time here. Or you can also have these beamline proposals where you can submit a proposal and the committee will evaluate. And like you can spend some weeks here doing your experiments, not only at the synchrotron laboratory, at the beamlines, but you can also use all the infrastructure, the support laboratories that we have here. But I think the short path is direct contact the researchers and show interests and informations and make this way as a collaborator.
Excellent. Thanks a lot. Any questions from this studio?
I've got one.
Yeah.
I've got a question about your micromodel. How did you manage to get the channel? Like what method did you use? Is it like chemical reaction or did you carve into the rock to make your microfluidics? And my second question would be, with the method that you use, whether you will be able to make an actual porous media, because currently you have just single channel. Would you have the opportunity and flexibility to make some pores and flows and make a more complex porous media?
Yeah. So the first question, I didn't give much details during the presentation because it's something that we are working at. So we got some results that we could do, but like it works for one sample and does not work for the other. But basically we used a drill, was a mechanical process using just water to refrigerate the rock. And we could do in some samples, and other samples that are more with bigger porosity or rocks a little bit more harder. It didn't work, and we need to figure how we are going to do it yet. And about the second question, yes, we are thinking on suit more complex structure. One path would be not draw the channel, but use the pores of the rock to make the fluid pass through. However, as we are yet interested in the mechanisms and to see if the rock has the capacity to absorb, to store the CO2, and as my project also involves other students that are working on computational simulations. So, as I mentioned, as controlled as possible, the experiment to be now, it's better for us. So that's why we decided to start just with the channel, with a controlled path to the fluid that we are sure that the fluids there is interacting. So for now, it's easy for us to understand, but for sure in the future, we are thinking in understanding the pore properties, how would be the micropore structures, micropore changes induced by the mineralization.
Excellent. Any more questions?
Other questions?
I have.
Yeah.
Okay.
So I've got a question about like the boundary condition in your system, because you're using at some one side you have glass or some way to kind of seal your porous media, but the other way, like it's a rock, and all other parts of the rock are exposed to basically atmosphere. So if you're, based on your title, you're gonna use carbon dioxide, right? And then so basically at some point carbon dioxide can infiltrate through the rock, and so basically the boundary condition that you're applying might not be exactly what you expect. So comment on that.
Yeah, for sure. So the point is that we are not using the supercritical carbon dioxide. We are using it dissolved in water. So it's the liquid carbon dioxide. And the flow test that we performed, I showed the movie in the presentation. So we saw with that on that flux of 0.5 millimeters per minute during the entire day, and we couldn't see any leaks. It was sealed with a Kapton tape, the channel, and we didn't see the leaks, neither on the tape on the channel or through the pores. So for now, the test that we're doing now with the liquid carbon dioxide, it is working very well when we are controlling the liquid carbon dioxide inside the channel.
Okay, thank you.
Fang, you had a question?
Yeah, just a quick follow-up question regarding the experimental method. So can you explain a little bit about how you sealed the rock to another material?
I think I came back my presentation, right?
Yeah.
You can see it, right? So here, where the channel is dropped, like in this surface, we have just a Kapton tape. It's a tape that is transparent to X-ray and is extensively used here in the experiments in the Synchrotron Beamline, because X-ray do not see it, so it's transparent. So it would interfere our experiment, and it's just glued here
I see.
inside the channel.
Thank you.
Excellent. Then I guess now it's time to wrap up this session, and we can again thank both of our speakers, Aluizio and Daphne, for their wonderful talks and for the introduction to this excellent synchrotron facility. Our next session would be on July 11th. We will have two States-based researchers, Zoe Canavas from the Morales Lab from the University of California, Davis. She will talk about flow phenomena under pore-scale spatial heterogeneity in geological media. And our second speaker will be Hangfang Cao from the Kong Lab in the University of Minnesota. And she will give a talk about density effects on the emergence of unstable focus flow in fractured systems. With that, I would like to close this session, and thank you all for joining us. And I hope that you will enjoy your summer vacations. This is our team. And if you want to suggest any speaker or you have any comment for us, please contact us at porosmediattt@gmail.com. Thank you and have a nice evening or morning, and ciao.